So the last video we're going to work on is the data collection for the pH procedure, and we're going to look at the effects of pH on our amylase enzyme reaction with starch. And so if you look at your table, we have 3 test tubes, and all 3 of them have amylase and starch inside of them because we know that this reaction does work, as we did discover in our enzyme specificity and our positive and negative control procedures. However, what we are changing in this procedure is the pH in which these reactions are going to be occurring. So in tube number 1, we have a pH of 2, which is very, very acidic, like stomach acid acidic. In tube number 2, we have a neutral pH of 7, a neutral pH of water, basically where our body is. And then in tube number 3, we have a very basic pH, which is a pH of 12. And so we're going to see, does amylase work well or poorly in these different pHs? So you're going to basically tell me the color, you're going to report the color, you're going to say, does the color indicate that there is sugar present or not? Because remember, again, Benedict starts out as this bright blue color, and we're going to be comparing, did it turn to orangey brown or not based on the sugar? And then based on that sugar being present or not, you can tell me, did the reaction happen between amylase and starch, right? Because starch is not a sugar, but amylase is breaking starch down into sugar called maltose. So here we have our 3, and I'm going to go ahead and show you 1, which is the acidic pH. And as you can see, the bottom is still kind of bluish green, and the overall color of the test tube, or sorry, of the contents of the tube, are this like pale, like yellow, a very weak color, somewhere between a blue and orange. And as you can see at the bottom, it's still kind of bluish with the starch mixture. So it's not an orange, it's not a brown, it is this kind of weird, like dirty yellow water color. In test tube number 2, we have our neutral pH, which was a 7, and we have this pretty bright orange carroty color. So you're going to record that down and tell me, what does that indicate as to whether or not sugars are present. And then in test tube number 3, we have the basic environment with a pH of 12. And as you can see, this one also has that bright carrot orange color in test tube number 3. So you're going to tell me, does that mean that there is sugar? Sorry, are there sugars present based on the Benedict's test? And if there are sugars present, tell me what happened with amylase and starch. And if there are no sugars present, tell me what happened with amylase and starch. And again, I will have you compare all 3 of them together just to tell you that yes, test tube number 1 is quite different from 2 and 3. So the acidic environment did have an effect more than maybe the neutral and the basic environment did. So afterwards, you are going to go ahead and answer some questions. And those questions can be found out of the way to skip the pages forward. So what effect did low pH, a pH of 2, have on the enzyme? Based on your results, did amylase digest starch at pH 4? Explain your answer. This should say 2. So if you can, you're going to switch this actually to say 2, right? Because our low one was a 2. And then what effect did high pH have on the enzyme? Right, that's the pH 12. And if digestion failed to occur in all test tubes, how might you test to see if the enzyme was permanently denatured or just temporarily inhibited? So is it permanently changed to the fact that when you adjust the conditions back into a working condition that you already know works, does it work again? Because if it does, then it's just temporarily inhibited, right? But if you go from an extreme pH and you go back to neutral where it's supposed to work, and we know that it works, and it doesn't, then it's probably permanently denatured, okay? So try to explain that for yourself in your own words, and then finish up your questions on the last page, which are the analysis questions, and then you will turn in your lab report.